Welcome back to biology. This is Mr. Kubuske, and this is biology. Today we're going to finish up talking about Unit 7, which is all about reproduction. We've talked about chromosomes, we've talked about mitosis, we've talked about meiosis, and now we're going to kind of tie all those together. We're going to talk about something called karyotyping, and actually a pretty interesting subject matter that goes along with karyotyping, which is genetic disorders. Now, just to warn you, I'm going to be showing some mature content, some pretty graphic images of some babies that have some of the genetic disorders that we're going to be referring to today. I just want to make sure that you are aware of it and that you can handle it in a mature manner. But you're all young adults. I expect you should be able to handle it pretty well. Okay. So again, we're talking about karyotyping and genetic disorders. Look for those key ideas as we go through here. Okay. All right, so how do we read chromosomes? That's the first question we're trying to answer. Like, we've got all these chromosomes, these 46 chromosomes in our diploid cells. What do we do with them? How do we use them? Actually, what we'll do is we're going to put them in order from biggest to smallest, autosomes first and the two little sex chromosomes last, and we're going to call that picture of our 23 chromosomes a karyotype. A karyotype is just a picture of the homologous chromosomes paired up. Again, we're going to go from autosomes, which are the chromosomes that control everything about your body, and then we're going to go biggest to smallest there, paired up, and then the last two we're going to put on there are the sex chromosomes. We're either going to put them homologous, which means XX, or non-homologous, which means XY, okay? But what do we do this for? Like, what's the point? Are we just doing it for fun? No, not really, okay? There's three purposes of a karyotype. The first one is obviously to study the differences in the chromosomes, the bigger ones, the smaller ones, where the stripes are, where the stripes aren't. Is the centromere high? Is the centromere low? What do they do? Like, why are certain ones smaller and groups of them bigger, so on and so forth, okay? We can identify gender. Obviously, we just talked about XX and XY. And then almost most importantly to a common person would be the fact that we can actually determine genetic disorders before a baby is ever born. So we will know, for example, if a baby is going to have Down syndrome before that baby is ever born. Okay, just something to think about. Okay, so let's take a look. This is a karyotype of an individual. This is a human being because we have 23 pairs of chromosomes. Now take a look at it. You notice anything wrong with it? Is this individual male? Is this individual female? Is this a diploid or haploid? What do you think? Okay. Now this picture, for example, is a female. How do I know? There are two X chromosomes and there's no Y chromosome. Okay, and then each one is paired up correctly. So this individual is a healthy female in a diploid cell. Okay. This karyotype, go ahead and take a look. Male or female? I know it is male because there's an X and there's a Y and everything's paired up correctly, so this is a healthy male. Lastly, look at this karyotype. Okay, this is a female. I know that because there's two X's, there's no Y's. But if you'll notice, we've got a problem here. There are 3 chromosomes where number 21 should be. Instead of everything being paired and only having 2, there's an extra little section of DNA. And believe it or not, just this one extra little section is what actually causes the syndrome we know as Down syndrome. Okay. So what causes genetic disorders like Down syndrome? It's a process called non-disjunction. If you break the word apart, you know that junction means where things come together or cross. Disjunction means they should be separated. Non-disjunction means it was supposed to separate, but it didn't. It moved all together. So a pair of sister chromatids, I need to change that actually, sister chromatids or homologous chromosomes, either one, do not separate the way that they should. Okay, and when does this separation occur? It occurs during anaphase 1 or anaphase 2. And then this can cause genetic disorders like birth defects or even miscarriages in some cases. Okay? So this is what should happen during anaphase 1. Anaphase 1, remember, we have duplicated chromosomes that are going to separate out. X's going to either side. Okay, the duplicated chromosomes going either way. During non-disjunction, that should happen. I should get a blue X's on the left and red X's on the right, but instead, one of them doesn't separate correctly, and I only get 1 duplicated chromosome on the left and then 3 on the right. Okay, the resulting cells are what we have down here at the bottom. So these 2 cells are missing a chromosome. These 2 cells have an extra chromosome. So no matter what, if you get one of these 4 cells, there's going to be some type of genetic disorder. If it occurs during anaphase 2, okay, the duplicated chromosomes, okay, the homologous chromosomes should separate normally. But during non-disjunction, one of the homologous pairs does not separate, and therefore one of the cells gets 1 chromosome and the other gets 3. Okay, and this is what they should look like at the end. Okay. So what causes these genetic disorders? What causes these problems? Okay. Well, the first problem is known as trisomy. Trisomy is when you get 3 sets of a chromosome. You're only supposed to get a pair, but instead you get 3. Monosomy is when you get only 1. So the trisomy goes one direction, monosomy would go the other. Sometimes we get something called a partial addition, where an extra piece of a chromosome breaks off and goes with the other homologous chromosome, or it could be a partial deletion. Okay, if you think about it, and I'll try to draw it big enough that you can see. I don't know if it's bright enough in here because I have all the lights on. So you can see it a little better. Okay, so let's say I had a homologous pair, okay, these 2 red ones, and they were supposed to separate, so I get one going left and one going right. Okay, but instead what happens is a little extra chunk of this goes with that one. So this one kind of looks like a P almost, and this one looks like the bottom of a lowercase I. Okay, so the one that has the P is a partial addition because it got an extra piece. The one that looks like an I is a partial deletion because it lost an extra piece. Okay, inversion and translocation. This actually goes back to when we talked about DNA mutations. We talked about chromosomal mutations as opposed to point mutations. So these are the chromosomal mutations. We're kind of going back to that now. It's interesting because it kind of all ties together. But inversion is where a piece of a chromosome flips, and then translocation is when a piece breaks off and goes to another chromosome and inserts itself. Okay? Now, again, I want to warn you one last time. We are going to be looking at some pictures of mature content here. Please be advised that we're going to be looking at those. Okay, and I want you to understand that the individuals that we're looking at, they have done nothing wrong. The parents have done nothing wrong. It's just that sometimes these chromosomes do not separate properly. Now, there's things that can cause that. If you go on to other biologies, you'll talk more about that. Like the age of the woman when she gets pregnant can cause more trisomies or monosomies. But we're not going to get into that in this class. You would do that more in like an AP Bio, IB Bio, or in your college biologies. Okay. So the first genetic disorder we're going to talk about is Down syndrome. I don't like to use the word popular, but this is the one that most people know of as being a genetic disorder. Okay. For each one of these, I want you to give me the name, and then I'm going to give you the cause. You can write down the symptoms, I guess is the word to use, or the problems, but you don't have to, and that's why they're going to be written in green here. Okay. But what actually causes Down syndrome? So if I look through the karyotype, I kind of go through it. I got a paired. I'm good. All these are paired up. Everything's good so far. And then I get right here to number 21, and boom, I have 3 instead of 2. So this is the problem. I have 47 chromosomes, which means I have a trisomy at number 21. Okay, so this is the important part. I want you to know what causes Down syndrome. Klinefelter Syndrome. Klinefelter Syndrome, believe it or not, and I'm not trying to start rumors or anything, but I've actually read some material on the subject, but many people believe that George Washington had Klinefelter Syndrome. Klinefelter Syndrome, it's a male, but they have like female secondary sex characteristics, meaning that they have immature breasts, they won't get facial hair, they'll get wide hips and narrow shoulders, things like that. And they're usually much taller and bigger than other males or females of that time. Like think about George Washington, you think of pictures of him and stories of him, they talk about how he was a hulking man, he was so much bigger than everyone else. And then if you see pictures of him, he always is kind of very, proud, very chesty. Well, I mean, that kind of leads us into the thought of that he might have Klinefelter syndrome. And then if you look at this part where it says he's usually sterile, if you think back to it, George and Martha Washington never actually had any children, and this may have been the reason why. So, but what Klinefelter syndrome actually is if you'll notice here, there are 2 X's and a Y. So therefore, this person is going to have, again, male and female characteristics when they go through puberty. So what actually causes the Klinefelter is they have 47 chromosomes, and it's an XXY. Okay, it's an XXY. Now you can, as people asked before, and we're not going to talk about it, but you can be an XXX. I don't know how to describe it. I don't have the words for it, but that could be a possibility. And then we'll talk about what happens if you just get an X here in a second. Okay, so Klinefelter syndrome, there's 2 causes of it. Female gives an X, but the male gives his X and Y, or the female gives both their X's and the male gives his Y. Okay, Turner syndrome, here is the one that's just an X. You don't get the second sex chromosome. So these individuals will be female, but they'll always be immature females. And what I mean by that is that they will not go through puberty. There's obviously some physical deformations as well, and then there's some mental retardation involved. Okay, so what causes Turner syndrome? This person only has 45 chromosomes, and we could call it monosomy 23, but technically we don't ever refer to the sex chromosomes as number the 23rd pair because they're not always paired, because one's an X and one's a Y sometimes. So a lot of times they'll just refer to Turner syndrome as X0 because there's not a second sex chromosome. Patau syndrome. Obviously, this is a pretty severe disorder. Deaf, cleft palate, heart problems, low survival rate. What actually causes it is right here. There's a trisomy at number, and all you have to do is count 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. So trisomy 13, so they have 47 chromosomes. Now, this is just a trisomy. Well, so is Down syndrome. So why do people with Down syndrome, or why are they able to lead healthy lives? Why are they able to be productive members of society? Whereas people with Patau syndrome will usually die within the first 3 months of birth. Well, the reason is, if you look, what's the difference between the 13th pair and the 21st pair? The 13th pair is a larger set of chromosomes than the 21st. So there's more DNA added, so there's more problems than there was at the 21st pair. Okay, so again, the further I go up, the higher up the chain, up the chromosomes, that these trisomies or monosomies occur, the more drastic the disorder, the more problems, and the less survival rate. A lot of times, if you get trisomies at the higher chromosomes, that baby will never develop. There'll be a miscarriage, and that baby will actually never even be born. Okay. Next one, Edwards syndrome, the trisomy 18 right here. So Edwards syndrome is right in between Patau's and Downs. So there are, again, low survival rate because these are, again, going to be larger chromosomes than the 21. But obviously you can see the problems aren't going to be as severe as Patau's. Now, obviously there's still some problems here, especially in the joints, clenched fists, disformation in the joints. You can see, I don't know if you can tell, but this individual's right leg right here bent all the way back behind his right arm. But again, a very low survival rate with individuals with Edwards syndrome. So again, 47 chromosomes and a trisomy 18. Very sad. And then the last one I'm going to want you to learn is something called cri du chat, which means cry of the cat. It's actually French for cry of the cat. And I'm only showing you chromosomes 4 and 5 here. So what's the problem? And someone might say, oh, well, there's an extra piece on this one. Well, actually, that's not true, because if you look, 4 and 5 should look similar. So the problem is that I'm missing a piece here. We call that partial deletion. It's called cry of the cat because people with cri du chat have a very high-pitched voices. I actually know someone with cri du chat. And again, it's very distinct. You can really tell that this voice and this larynx is disformed because it's such a high-pitched voice, and that's where the phrase cry of the cat comes from. There are mental handicaps associated with cri du chat as well. Okay. So, how do we read a karyotype? We're going to be doing that for the next day or so in class. What do we do with this karyotype? What are the things that we're looking for? Well, first thing you got to ask yourself is, are there 46 chromosomes? If there's more or less, you know you've got a problem. So if you want to count them each individually, you can. That would be a way to tell. Or you should know that they should come in pairs. For humans, there should be 23 pairs. If something's not paired correctly, okay, you know there's a problem, especially if it's in the sex chromosomes or in the higher number chromosomes. Okay, and then number 3, are there any rearrangements? Were there any additions or deletions? Like my pie back here, okay, the big P with the extra piece and the little I, okay? That's the next thing you need to look for. Did things get shifted around? Did the bands match up and that type of stuff? So those are the things you need to look for when you're reading a karyotype, okay? I appreciate your sensitivity on this subject. I know a lot of you out there probably know someone with one of these disorders, and so I hope that this is kind of a refresher. I hope you know that they didn't do anything wrong. It's just it just happened that in that sperm and in that egg cell, when they went through their meiosis, just something didn't get disconnected correctly, and unfortunately, it does happen, and it's sad, but it is a part of life. So again, I hope you learned something. If you have further questions, please research it. There's tons of information on these different syndromes, and I only covered 6. There are dozens of these syndromes that you could learn about out there, and I'm sure some of you have some other stories about some of the other ones. But if you have questions, again, contact me, visit the website, or follow me on Twitter at Coach Kubuske. Hope you learned something today. Have a great day, and stay warm.